For most of human history, we looked at each other and saw difference. We saw the color of skin, the shape of eyes, the curl of hair, and we built entire worlds around those differences. We built laws, borders, hierarchies, and even sciences around the idea that beneath those visible features lay something deeper, something biological, something that divided us into fundamentally separate categories of human being. And then science caught up. And what it found didn't just challenge that idea. It dismantled it piece by piece at the most fundamental level possible, inside the molecule that makes us who we are. This is the story of what happens when you actually compare the DNA of human populations from every corner of the earth. Not what people assumed would be found, not what ideologues on any side hoped for, but what the data, the cold, patient, unambiguous data, actually revealed. And the answer is one of the most profound things science has ever uncovered about our species. It begins, as most great stories do, with a question. A simple one on the surface, but loaded underneath with centuries of assumption. The question was this. If you take human beings from vastly different parts of the world, from sub-Saharan Africa, from East Asia, from Scandinavia, from the indigenous populations of the Amazon, and you look inside their cells, inside the very instructions that build and run a human body, what do you find? Are humans divided at the genetic level, the way our social categories have always suggested, or is something else going on entirely? To understand why this question matters so much, you have to understand what DNA actually is and what it does. Every living cell in your body contains a complete copy of your genome, a sequence of roughly 3 billion chemical letters arranged in a specific order that serves as the instruction manual for building you. Those letters, called nucleotides, come in 4 types, and it is the order in which they appear that determines everything from the color of your eyes to how your immune system fights off a virus. The genome is essentially a biological text, and every human being on Earth carries a version of that text inside every one of their cells. Now here is the first extraordinary thing. When scientists began comparing those texts across populations, across what we would socially call different races, the first number that emerged stopped researchers in their tracks. Human beings, all human beings, regardless of where their ancestors came from, regardless of what they look like on the outside, share 99.9% of their DNA. Not 90%, not 95%, 99.9%. The genetic difference between a person from Nigeria and a person from Norway, between someone whose family has lived in Beijing for 1,000 years and someone whose family has lived in Peru, is, at the level of the genome, almost non-existent. We are, in the most literal biological sense, virtually the same. That 0.1% that differs, the variation that does exist across human genomes, is where the real story gets interesting. Because when scientists began to study that variation carefully, they discovered something that turned the popular understanding of race completely on its head. You might expect, if races were genuinely distinct biological categories, that the major genetic differences would fall neatly between groups. You would expect European genomes to cluster tightly together and differ substantially from African genomes, which would differ substantially from Asian genomes, and so on. That is what a world of biologically distinct races would look like in the data. But that is not what the data shows, not even close. What researchers found, consistently across study after study, was that the overwhelming majority of human genetic variation, roughly 85% of it, exists not between populations, but within them. Within a single village in West Africa, within a single neighborhood in Tokyo, within a single town in rural Sweden, you find more genetic diversity than you find when you compare those groups to each other. The variation that exists between what we call racial groups accounts for only about 15% of all human genetic difference. Think about what that means for a moment. The concept of race, as it has been used for centuries to divide humanity, to justify hierarchies and laws and violence, maps onto a portion of human genetic variation so small that it is almost biologically irrelevant. This finding was not the product of one study or one team of scientists with an agenda. It has been replicated across decades of research, using increasingly powerful tools, in larger and larger data sets, with greater and greater precision. The Human Genome Project, completed in 2003, made it definitively clear. The Wellcome Sanger Institute, working with the University of Cambridge, sequenced 929 human genomes from 54 geographically, linguistically, and culturally diverse populations from around the world, using the most advanced sequencing technology available. The Genome Aggregation Database Consortium, known as GNOME-AD, assembled genetic data from more than 125,000 individuals spanning populations across Europe, East and South Asia, Africa, and beyond, cataloging 241 million genetic variants in the process. Every time researchers have gone back to this question with better tools, the answer has come back the same. There are no clear genetic lines separating the groups we call races. There are no genetic variants that appear in all Europeans and no Africans, or all Asians and no one else. When scientists look at the actual distribution of genetic variation across the human species, they find something that looks less like a set of distinct boxes and more like a continuous gradient, a spectrum of overlapping variation shaped by geography, history, and movement, not by the racial categories that human societies invented. To understand why this is the case, you have to understand where we came from. And this is where the story of human genetics becomes genuinely breathtaking, because the history written in our DNA is not the history most of us were taught. Modern humans, Homo sapiens, did not spring up in multiple places around the world simultaneously, each group developing independently and distinctly. The evidence now is overwhelming that our species originated in Africa and that all human populations alive today descend from those ancestors. But even that statement turns out to be more complicated and more wondrous than it sounds. Recent genetic research, including a sweeping study that made headlines in 2026 by analyzing the genomes of diverse modern African groups, has found that early human origins were not a single spark in one location, but rather an intricate intertwining of multiple populations spread across the African continent. These groups diverged and reconnected, mixed and separated over hundreds of thousands of years, exchanging genes and cultures across vast distances long before any human being set foot outside Africa. What this means is that even the story of human origins is a story of mixing, not separation. The deep roots of our species are entangled, not branching cleanly apart. And the genetic legacy of that entanglement is still with us, written into the DNA of every person alive today. When some of those populations eventually migrated out of Africa, a process that began somewhere around 60,000 to 70,000 years ago, they carried a sample of African genetic diversity with them. A sample, not the whole. This is crucial. The populations that left Africa took a subset of the genetic variation that existed in Africa, and as they spread across Asia, Europe, the Pacific, and eventually the Americas, they diversified further, accumulated new mutations, and adapted to new environments. But because they started with a sample of African diversity, the populations that remained in Africa retained significantly more genetic variation than any other population on Earth. Africa is, genetically speaking, the most diverse place on the planet, not the least diverse, not the most exotic or most different, the most internally varied, the most genetically rich, the deepest well of human biological history. Two people from neighboring villages in the Democratic Republic of Congo may be more genetically distinct from each other than either of them is from a person in Ireland. This is not a metaphor or an approximation. It is a direct consequence of the fact that human beings have been in Africa for far longer than they have been anywhere else, and diversity accumulates with time. The Wellcome Sanger study found exactly this. When researchers dug into the genomes of populations from southern Africa and central Africa, they discovered enormous quantities of previously undocumented genetic variation, variants that had never appeared in any study before, because previous studies had so dramatically underrepresented African populations. The genetic complexity of Africa was not just larger than expected. It was larger than the rest of the world's genetic complexity combined. And yet, for most of the history of genetic research, Africa was treated as an afterthought. The vast majority of large-scale genetic studies, the studies that shaped our understanding of disease risk, drug response, evolutionary history, and human biology, were conducted overwhelmingly on people of European descent. The NOMAD consortium was blunt about this. Most genetic studies of disease have been conducted in European ancestry cohorts, and the results often do not transfer well to other populations. What this means in practice is that the tools used to predict disease risk, to calibrate medication doses, to screen for genetic disorders, have been calibrated on one narrow slice of human genetic diversity and then applied, often incorrectly, to the whole species. The scientific blind spot, created by ignoring non-European populations, has not just been an intellectual failure. It has had real consequences for the health of billions of people. Now back to the question of what makes different human populations look different. If 85% of human genetic variation exists within populations rather than between them, and if there are no clear genetic dividing lines between racial groups, then what exactly is happening when we see differences in skin color, facial features, and body type? This is where genetics becomes particularly elegant. The visible traits that we use to assign people to racial categories are controlled by a remarkably small number of genes, most of them responding to a single environmental pressure, the amount of ultraviolet radiation in the environment. Skin color, which is the primary visual cue human societies have used to construct race, is a relatively recent and geographically variable adaptation. Populations whose ancestors lived near the equator, where ultraviolet radiation is intense, developed higher concentrations of melanin as a protective adaptation. Populations whose ancestors lived at higher latitudes, where sunlight is weaker and vitamin D synthesis is a greater concern, developed lower melanin concentrations. This adaptation happened multiple times, independently, in different populations. and it happened relatively recently in evolutionary terms. The genetic variants responsible for light skin in European populations are different from those responsible for light skin in East Asian populations. Skin color is not a deep biological marker of ancestry. It is a superficial, adaptive response to sunlight, as genetically shallow as it is visually prominent. The same logic applies to other visible traits. The epicanthal fold in East Asian eyes, the textured hair of African populations, the variation in nose shape across populations, these are all adaptations to local environments. Controlled by small numbers of genes, they tell you almost nothing about the deeper structure of a person's genome. Beneath those visible differences, the genomes of people from radically different parts of the world are overwhelmingly, staggeringly similar. What the science is telling us, with a clarity that has only grown over decades of increasingly sophisticated research, is that the concept of biological race, the idea that humanity is divided into discrete genetic categories that correspond to our social racial classifications, does not exist in the genome. The National Academies of Sciences, Engineering, and Medicine stated this explicitly in a landmark 2025 report. Race is a socio-political construct, not a biologically valid taxonomy for genomics research. The differences in health outcomes, life expectancy, educational achievement, and economic well-being that exist between racial groups and societies around the world are not explained by genetics. They are explained by history, by the structures that societies built on the false premise of biological race, by the compounding effects of discrimination, unequal access to health care, unequal educational resources, and the economic legacies of slavery, colonialism, and segregation. This is not a politically motivated conclusion smuggled into science. It is what the data says. It is what 70 years of increasingly rigorous population genetics research says. The leading population geneticists in the world are clear about this. As one prominent researcher put it, the narrative that there are discrete genetic racial clusters is not what the data tells us, and it is not what the last 7 decades of research tell us. But here's where the story becomes more nuanced, and nuance matters enormously in science. Saying that biological race doesn't exist is not the same as saying that population ancestry is biologically irrelevant. These are two very different things, and confusing them does real harm. Genetic ancestry, where your ancestors came from geographically, is real, it is detectable, and it does have some biological implications. Because human populations were relatively isolated from each other throughout much of their history before the great mixing events of the last few centuries, genetic variants do cluster geographically. If you sequence someone's genome, you can often make a reasonable inference about the broad geographic region their recent ancestors came from. Some genetic variants that affect disease risk are more common in certain geographic populations than others. Sickle cell trait, for example, is more common in populations whose ancestors lived in malaria endemic regions, because it confers some protection against malaria. The BRCA1 and BRCA2 variants associated with elevated breast cancer risk are more common in people of Ashkenazi Jewish descent. These are real patterns with real medical relevance. But, and this is critical, geographic ancestry is not race. The populations that share certain ancestry-linked genetic variants do not correspond to the racial categories that human societies have constructed. Sickle cell trait is common in sub-Saharan Africa, but it is also common in parts of the Middle East, the Mediterranean, and South Asia, populations that are not socially categorized as the same race. The racial category of black in the United States encompasses people with ancestries from dozens of distinct African populations that are themselves enormously genetically diverse. Using race as a proxy for genetic ancestry in medical research or clinical practice introduces errors, not precision. This distinction between the real, medically relevant concept of genetic ancestry and the scientifically invalid concept of biological race is one of the most important ideas in contemporary genomics. And it is one that is too often lost in popular discussions of the topic. There's another chapter of the human genetic story that is worth understanding because it adds yet another layer of complexity to the idea of pure racial categories. And that is the story of ancient mixing, the genetic legacy of encounters between Homo sapiens and other human species that lived alongside us for tens of thousands of years. When modern humans migrated out of Africa and spread across Eurasia, they encountered populations of Neanderthals who had been living in Europe and Western Asia for hundreds of thousands of years. They also encountered a group called the Denisovans in Eastern Asia, and they did what humans have always done when they met other people. They mixed. The Wellcome Sanger study confirmed what earlier research had suggested. The genomes of present-day human populations carry the genetic signatures of those ancient encounters. Most people alive today whose ancestry is not exclusively sub-Saharan African carry somewhere between 1% and 4% Neanderthal DNA. People with ancestry from Southeast Asia and Oceania carry additional Denisovan DNA. Indigenous Australians and Papua New Guineans carry particularly high levels of Denisovan ancestry. What this means is that the human species is not and has never been, a set of pure, separate lineages. We have been mixing since before we were fully modern. We mixed with Neanderthals. We mixed with Denisovans. We mixed with each other across every continent in every era of history. The genome of every person alive today is a palimpsest, a document written and rewritten over hundreds of thousands of years of movement and encounter and contact. The idea of racial purity, which has caused so much suffering throughout history, has no basis whatsoever in genetics. It never did. The data has always said the same thing. We just needed the tools to read it. What is perhaps most remarkable about all of this is not just what the science reveals about human genetics, but what it reveals about human history. The DNA data, when you read it carefully, tells a story of constant movement, constant encounter, constant mixing. From the deep African origins of our species, through the migrations that populated every continent, through the ancient encounters with Neanderthals and Denisovans, through the historical migrations and trade routes and invasions and diasporas of the last 10,000 years. The human genome is a record of relentless human restlessness and connection. The Sanger study found deep and gradual population separations within Africa, contrasting histories between hunter-gatherer and agricultural groups over the last 10,000 years. It found the genetic signatures of the great out-of-Africa migration. It found evidence of the settling of the Americas, of the Polynesian expansion across the Pacific, of the spread of agriculture from the Fertile Crescent into Europe and from China into Southeast Asia. Every great movement of human history is recorded somewhere in the genome. And in all of that data, through all of that history, what you find is not the story of separate races diverging from each other in isolation. You find the story of a single species constantly interacting, constantly exchanging genes and ideas and culture, constantly becoming more interconnected, even as geography and time created local differences. The genetic differences between human populations are real. They exist. They have some biological significance. But they are the differences of branches on a single tree, not roots of different trees entirely. We are, as the data so clearly shows, one people. Not in a sentimental or wishful sense, but in a hard, empirical, genomic sense. The 0.1% of our DNA that varies between individuals encodes local adaptations, ancestral histories, and the signatures of ancient encounters, but it does not encode separate categories of human being. There is no genetic marker for race. There is no genomic sequence that says this person is fundamentally different from that person in any way that corresponds to our social classifications. The molecule itself refuses to cooperate with the story we told ourselves. Now, some people, when they hear all of this, feel a kind of intellectual vertigo. If race is not biology, why does it feel so real? Why do we see it everywhere? Why does it matter so much in the lived experience of billions of people? The answer is that things can be socially real without being biologically real. Race is one of the most powerful social forces in human history. It has shaped economies, governments, wars, laws, art, science, and personal identity for centuries. The fact that it has no foundation in genetics does not make it disappear as a social phenomenon. It makes the social phenomenon more remarkable, that we built such enormous structures on such a thin biological foundation. And this is perhaps where the science is most useful, not in dismissing the reality of race as a lived experience, but in correctly identifying what it is. Race is something that societies create and enforce and reproduce through culture and law and practice and belief. It is not something written into the genome at conception. Understanding that distinction is not just intellectually important, it is practically crucial, because it tells us something about where the solutions to race-based inequality actually lie. They do not lie in biology because the problem was never biological. They lie in the structures and systems and beliefs that human beings created, which means they are structures and systems and beliefs that human beings can also change. The science of human genetics has given us something genuinely extraordinary, a clear, empirical answer to one of the oldest questions in human history. Are we fundamentally different from each other? Are the categories we use to divide ourselves real at the deepest level of our biology? The answer is no. The answer, written in 929 sequenced genomes and 241 million cataloged variants and decades of careful, rigorous, replicated science, is no. We are one species with one origin, shaped by one shared evolutionary history, carrying one genome that varies continuously and beautifully across a single interconnected human family. The visible differences between us are real. They are interesting. They tell stories of adaptation and migration and local history. But they do not divide us into separate biological categories. They are the surface expression of a species that has been everywhere, survived everything, and mixed with everything it encountered. There is something almost humbling about this when you sit with it long enough. The molecule that encodes every one of us, that carries the instructions for every human life, does not recognize the lines we drew on maps. It does not respect the borders we erected or the hierarchies we constructed. It tells a story that is simultaneously simpler and more profound than anything our social categories ever captured. It tells the story of a single ancient, restless, endlessly curious species walking out of Africa and across every corner of the earth, carrying its diversity not between groups but within them, leaving traces of every encounter in every genome, arriving in the present moment as 8 billion slightly different versions of the same essential thing. Scientists compared the DNA of human populations from around the world. And what they found was not evidence for the divisions we assumed. What they found was evidence for the most fundamental unity imaginable. Not the unity of identical copies, but the unity of a single family so old, so widely traveled, and so thoroughly mixed that no gene, no chromosome, no sequence of 3 billion letters can tell you where one race ends and another begins. Because that line was never there. It was always only in our minds. That is what the data says. That is what the science shows. And that perhaps is the most important thing genetics has ever told us about what it means to be human.